Hello, my name's Arthur Penty, and today I'm going to talk about artificial life in materio. So, artificial life in materio, this is a new kind of ALife we want to introduce, where we look at emergent lifelike phenomena within a physical material substrate. And we're inspired by material computing, which is exploiting naturally occurring physics for efficient computation, and we're inspired by evolution in materio, which is where you interface some evolutionary algorithm with a material directly in order to tune its behavior. And so one curious thing about this ALife in materio is that although the creatures exist within a physical material, the creatures themselves do not need to be physical. So this sounds kind of interesting but nebulous. So we'll look at a concrete example to understand this better. So we're going to look at something called artificial spin ice. This is our favorite system to work with. And this is just a set of nanomagnets arranged in some pattern. And on the left, we've got an example of a microscope image of them. And on the right, we can see some simulated dynamics. And each nanomagnet has what we call a binary spin. So it's either pointing along its axis in one direction, or it can flip, given enough encouragement, and point along the other direction. And so these magnets on the right, we can see they start polarized with the up magnets in green and right magnets in red. But we could apply some global fields or some other kind of stimulus, and this could cause a more interesting pattern to appear. And artificial spin ice are what's known as metamaterials, which means although we have many components, they all work together, and the emergent behavior is similar to that of a regular material. Artificial spin ice are of interest to physicists and computer scientists for all sorts of reasons. But today we're talking about ALife. So our main question is, where are the creatures? So we can start off with spin ice polarized down and left, as we can see from our color key here. And we can initialize some pattern in the middle. So we can think of these blue and purple magnets being in the off state, and then as flipping some magnets in the middle to turn them on. We can simplify this visualization by going to a grid-based view. We don't need to draw all the magnets. And also, we're not so interested in these background magnets, so we can give them a darker color so we can focus on what we've initialized in the middle. And so then we can start applying some global fields to these patterns, and we can see here it's not so interesting, it just kind of spreads out. But in previous work, we've shown if we choose a specific initial pattern, and then we use these fields here shown on the right, and we go through them in this order, A, B, B, then we get this nice creature-like behavior. And so we call this thing here the snake, because it kind of slithers along the plane like a snake. And if rather than applying one field and then taking a picture, as we're doing here, if we apply all four fields in sequence and then observe the system, we see this kind of translational behavior. And so we found these snakes because we were trying to find some analogy to the glider of the game of life in our magnetic system. And we were able to implement these snakes in the real world. So we produced some ASI in our lab, and we were able to initialize snakes on the right and have them move leftwards. And we can see here they fizzle out after a bit of time, but this is just due to the level of imperfections in our samples that we're able to create at our lab. So returning to the starting point, we can see in the snake, we have a creature that exists in a physical material, this ASI. But if we follow one of these snakes, we can see as it moves, none of the magnets are moving. The snake moves through the magnets. And so in this way, the snake is embodied, not in magnets, but in the state of the magnets. And this brings us to an interesting question. We know in ALife, we have these categories, hard, soft, and wet ALife. And where do our ALife in materio creatures fit into this? Well, in this example, we can easily discount wet because these magnets don't have any biological or chemical components. Soft ALife, you could argue here on the left, because we're simulating it, it could be soft. But this is a physical simulator, and of course the goal is to have it eventually in a physical ASI, so we can discount soft. And finally, for hard ALife, this typically refers to things like robots, either swarm robots or larger robots. And the emphasis is on hard agents or hard creatures, I think. And so here we don't have hard creatures, we just have a hard environment for them to live in. So we would argue that we're also not in this hard category either, and that perhaps we're a new kind of ALife. So we've seen so far, we know we can make one kind of glider in ASI, the snake. But ideally for ALife in-materio, it would be nice if we could find a diverse set of different life creatures. So we're going to look for still life, which means things like this that just stay the same. Oscillators, which have some kind of periodic behavior. And then these spaceships or gliders, which maintain their shape and move through the space. And we're going to use something called the evolutionary algorithm to try and find new creatures. And what this means is we will give some parameters to our algorithm to tune and give it some objective function to try and maximize. So we're going to give the EA control of initial a region in the center of the ASI. And here it can choose the starting pattern. And this is a 10 by 10 area of magnets. We're then going to apply a sequence of clocking fields, so AA, BB, CC, DD. And the A and B fields are just the ones we used earlier for the snake. And then we have these 4 new C and D fields, which are just the mirror image of the A and B fields. And so we're going to fix this clock cycle, but we will allow the EA to control the field strengths that are applied. And so we have 4 different field strength parameters. For example, H, small a, small b, controls the field strength of the small a and the small b fields, and so on for the other 3. And so in this way, we parameterize our 8 fields with 4 parameters. And so if there's one field the EA doesn't want to apply, it can just choose a very low field strength for that particular field. As well as giving it control of parameters, we also need to specify a fitness function so it knows what it's trying to optimize for. And we're going to use this kind of evolutionary method called map elites, which is where we specify a fitness function and then also some behavioral dimensions. So our fitness function is going to be homeostasis, so this idea of self-regulation, trying to maintain some kind of similar functioning. And we're going to simplify this concept of homeostasis down to the idea of just trying to stay the same size, which we'll count by counting the number of magnets in the on state. For our behavioral dimensions, these are the things which we're going to try and find diversity in. So these are ways to set up kind of niches for the algorithm to find. So one axis will be size. So we want to find a diverse set of different sized creatures. And on our other axis, we're going to have cycle length. And so this is just looking at how often we see a repeat in the dynamics of the creatures. So a cycle length of 1 indicates we've got still life because it's repeating every time step after some transient period. If we have between 2 and 19 cycle length, that means we've got some oscillatory behavior. And if we see a cycle length of 20, that's the maximum because we're running 20 steps of the simulator. And so 20 means it's a new state every time. So this could be a glider, or it could be just something that changes state every step. But in conjunction with our homeostasis fitness function, something that changes every step and remains the same size is quite likely to lead towards glider behavior. But it could find something else too. The last thing to talk about is cheat prevention. So we know evolutionary algorithms really like to find loopholes and ways to cheat. So we're going to try and preempt some of that now. One obvious way to cheat, if we're looking for still life, would just be use really low field strengths, so they don't have any effect on the system and it just remains frozen. So to combat that, we're going to require there to be some activity in the system. So even for still life, there must be some activity in the creature and then it can freeze into this still life behavior. Another kind of a bit more complicated cheating is this thing we see in preliminary testing, and that's this idea of one of the ways it can achieve oscillatory behavior is for the creature to very quickly grow to cover the whole array to reach the edges. And at the edges, we have this fixed boundary condition. And the creatures are able to abuse the fact that the edges don't switch to kind of self-regulate and make it easier to get this oscillatory behavior, kind of balancing the growth and shrinking. But we want the creature to kind of learn to do this itself rather than abuse the edges. So we're going to penalize it if it reaches the edge. Additionally, these are not really cheating, but we're going to penalize creatures that die. So all the magnets turn off. and we're going to penalize creatures that explode, so when lots of magnets turn on very quickly. So now we can look at the outcome of running this evolutionary algorithm. I'll explain what the propagator means in a minute, but first we'll start by looking at still life. So the still life is as we'd expect. There's some activity at the start, as we've necessitated, but then it freezes. And the smaller still life is not that exciting, but what's kind of interesting about the still life at the larger scales is, as our initialization region was 10 by 10, meaning 100 magnets, to get a still life larger than 100, it first needs to grow a bit outside of this initialization region and then freeze. And so the still life here is a bit more exciting because it has this two-stage behavior of growing and then kind of self-regulation and freezing. Next, we can look at some oscillators. So we see that oscillators don't span as much of the space as the still life, but we found a range of different periods. So I've written the periods in numbers onto the oscillators, and we can look at some of the period 2 oscillators first. So this is the smallest oscillator we found, and you see it's interesting. It looks kind of like it's two creatures that are kind of holding each other in balance. We then have some more small still life oscillators, and we've named some of the creatures that are particularly interesting, or just if we think they look like something. So here we have the crotchet and the spin wave, and then we have some more period 2 oscillators, and here's the largest 2-period oscillator. We can look at the 4-period oscillators. They all look quite similar. This one here we've named the frog because my colleague thought it looked a bit like a frog jumping and then falling back down. The 3-period oscillator is interesting because this is the only odd period oscillator we found. And again, my colleague said it looked a bit like a horse, so we've called it the horse. And I think this is a particularly nice name because you have this kind of triplet feeling to how a horse runs, so that's quite a nice name for it, I think. We can look then at the longest oscillator we found. This is the spaghetti. So we can see it. There's some initialization here, and then it gets into this long 14-period oscillator, where it kind of looks like these. It's many creatures bumping into each other to create this long period. The 10 and 12 look quite similar to each other, but quite different to anything else we've seen. They oscillate by having this growing and then shrinking behavior, and it looks quite complex. We can then look at some of the gliders. So I've called this section propagators because these circles here, these are all the gliders. And above this point, we didn't find gliders. We found something else, which I'll talk about when we get there. But we found the snake, as expected, a little length 2 snake. And we also found some other more strange gliders. So this one's going down and left after some initialization. And then these two are going up and right. We found this thing called the manta ray. And this is quite interesting because it goes, unlike the snake and these other gliders, which travel along the direction of magnetization or parallel to it, this travels perpendicular to the magnetization of the creature. So that's kind of cool. And we also have this thing called the plow, which is the largest glider we found. And what's interesting here is we have this kind of filled-in domain, which a physicist would tell you is a ferromagnetic ordering. And we have this kind of more sparse checkerboard domain, which is antiferromagnetic ordering. And so it's interesting, these two different types of ordering are working together here to create this creature. So above this size, we didn't find any gliders, but we did find things that had cycle length 20, so things that change every step. And the way they were achieved is by this growing. So it's a new state every step because it's growing. And again, we had this fitness function that was trying to have homeostasis, so minimize change. And so we can see while it's not able to be completely homeostatic, it's trying to minimize the amount it grows. So you get these kind of arms or tendrils reaching out. But we found lots of different creatures. And so one thing to remember is we're not just finding these patterns, the initialization patterns, but we're also tuning field strengths in order to get these creatures to work. And so one way I like to think about this is we find these different patterns, which are the creatures, and then the field strengths we use to drive these different creatures, these we can think of as being like the laws of physics in the universe the creature inhabits. And so we could take a creature, we could change the field strengths, and that's like moving it into a different universe and seeing is it able to survive and how does its behavior change in this other universe. And we can think about all these different universes we've found. Each of these creatures we've discovered are in their own universe. And we can describe a universe as this product of our 4 field strengths. And one way to analyze these different universes is to look at how these different field strengths change on average over the course of their evolutionary algorithm. And so this is the mean field strength in the population over each of these different fields. And we see the large AB field has the highest strength quite quickly. This gets very high. And the small AB field also is relatively high compared to the other fields. And so this seems to indicate that perhaps the C and D fields are not so important. And actually, further analysis reveals that for the vast majority of the creatures, they're not actually using these C and D fields at all. They're just using the A and B fields. And so if we discard the creatures that use the C and D fields, it makes the analysis much more tractable. So we'll do that for now. Sadly, we lose some of these creatures, so we lose the 3 and 4 period oscillators. But this allows us to make this 2-dimensional map of the different universes, and we can color them based on the creature we found in them. So these red ones are the still life universes. We can see they exist at the lower field strengths, which makes sense. Then we have these dark blue universes are the glider universes, and we can see this generally spans everywhere there are creatures. And lastly, the oscillators are shown in pink to cyan, and these we can see all exist up here in this top right corner. And so one interesting question is, if we have some creatures that live in universes that are nearby, can they visit each other? Can we put them in the same universe and see if they coexist? So we're going to define this region here, and we're going to simulate all the creatures in this region in one compromise universe with the same field strength to see what happens. And when we do that, what we find is we get these 7 different kinds of behavior besides the creatures that just die or explode when we move them into this compromise universe. We find 5 different gliders, and we find 2 period oscillators. And these are different oscillators here. And what's nice about this is that all of these creatures could then coexist in the same spin ice under the same fields, and they're able to meet each other and interact. So to conclude, this has been an introduction to ALife, in-materio, where we've tried to find lifelike behavior using only simple physical interactions within a material. And we found a diverse set of different virtual magnetic creatures. And in doing ALife, in-materio, we're trying to find lifelike behavior just arising from simple physics innate to a different material, and trying not to predefine our notions of what mechanisms are required for life, but more take this phenomenological view of observing things and seeing if they appear lifelike. So there's lots of future work to think about. An obvious missing aspect of life is replication. So could we find something either like a Gosper gun that shoots out gliders, or even something like a creature that's able to self-replicate itself? We saw the snake gliders had some trouble in the lab due to noise, but it could be that some of these other larger gliders are more robust. So that would be interesting to look into. We know in the game of life, we can use these gliders and structures to build machines and computers. So it would be interesting to see if we're able to put some of our creatures to work in a more functional way as well. And finally, we've shown a very designed approach, designing these creatures for different behaviors. But we could imagine just randomly initializing a large ASI with some clever fields and seeing if we can get some primordial soup and see what emerges from that. And actually, we've done some initial studies into this already, and it seems quite promising. So this is certainly something we want to look further into. But that is my talk. Thank you for listening. If you want to look at these creatures yourself, we have this online interactive version of the chart. But that's my talk. Thank you.
Good day, everyone. I am Nitirak Rayabphand from the University of Bristol. Today, I would like to talk about my work, a rapid parallel fabrication and control of diverse micro-robotic structures with magnetic functionalization. We all know that morphology impacts the behavior of the animal. Their bodies and limbs make them behave differently. We can study behavior of animals by building robots that look exactly like them. But the question is how to build those robots. The coming of rapid prototyping technology helps in building those robots within a short time, but still, current technologies such as 3D printing and lithography are time-consuming, lack in scalability, and still require many pre- and post-processing. Here from the background I have mentioned, they lead to some questions like how to fabricate the micro robot with the complex shapes, how to simplify the fabrication method, how to reduce the fabrication time, and how to scale up the fabrication process. Before we start, I would like to mention and I would like to introduce to the device that we have been working on for the micro robot fabrication. It's called the Macadome. The Macadome stands for the Magnetic Dynamic Optical Microenvironment. It is an open platform for high-resolution light-based and magnetic contactless for fabrication of hydrogel-based microstructures. The Macadome includes three systems: the projection system, the magnetic system, and the observation system. The projection system is made of the high-resolution near-UV light projector, which can project 4 or 5 nanometer wavelength to kill liquid hydrogel to be gelatin structures. With the high resolution, this allows to project down to 30 microns for a single pixel. The magnetic system includes four electromagnets aligned perpendicular to each other until 55 degrees from the base. This configuration can generate the magnetic field up to 3.5 millitesla in three-dimensional space. And the observation system is built of a high-quality camera and replaceable microscope lens. All of the structural part of the megadome are able to be printed by the 3D printer. This device is open and all the STL file for 3D printing are available on the link below. Before we step to the fabrication process, we have to prepare the hydrogel solution as it is the best material for the microstructures. The solution consists of first the gel, which is the biocompatible hydrogel that normally being in a gel state in low temperature and liquefy in the high temperature. And the next one is the LAP, which is the photo initiator. This provides a photo cross-linkable property to the gel. This property allows the gel to be cured from the liquid to be the gel state when it is exposed to the near UV light. So with the gel, with the LAP, the gel will not turn back to the liquid again in a high temperature after the photo cross-linking process. And the last one is the IOP or the iron oxide particles. Adding these particles to the gel will provide a magnetic property that allow the microstructures to be controlled or manipulated by the magnetic field. And here we can go by the fabrication process. First, we need to prepare all of the material used. We need to prepare the gel solution using the material I have mentioned before. And then we transfer the solution into the petri dish and then move the petri dish into the Macedon. And then we use the magnetic oscillation to distribute all the IOPs around the solution. And when all the IOP is distributed around the solution, we align the IOP in the uniform direction using the uniform magnetic field, and then we fix it. And then the image pattern is projected to the solution using the high resolution projector that we have. This one will induce the photo cross-linking process to the Gemma solution and kill the structures. After 45 seconds of curing, we wash out the uncured solution and then we transfer the micro robot after washing to a new petri dish for further experiment like the manipulation experiment by the magnetic field. In this work, we intend to prove that we can build the microstructures in different shapes and size in parallel. Here are the images we use as the pattern for the fabrication. The white area is the area that the projector display the shape, and the black area is the area with no projection. For the simple microstructures, we use the geometric shapes such as triangle, square, star, and circle as a pattern image. And for the complex structures, we use the fish-like and the tadpole-like as a pattern image. Here are the results of the microstructures after the fabrication. On the left-hand side image, the geometric structures were fabricated in different size and shape in parallel, and all of these were fabricated in a single batch. Also, the same to the right one, the fish and the tadpole were also fabricated in parallel, and all of these were fabricated in a single batch as well. And now let's take a closer look on the fabricated structures under the microscope observation. The scale bar we use for this observation is 500 micron. All the structures were fabricated using approximately 50 by 50 pixels image. The real size of the structures is approximately 2.5 to 3 millimeters. In overall, all of the structure have no severe damage and distortion appeared, and the IOP were clearly observed and the alignment is preserved within the structures. And when we overlay the pattern image onto the structures, we can see that the structure size is very close to the pattern image. There are some size shift in more complex structures, such as the edge of the star, the fin of the fish, and the tail of the tadpole. But in overall, the sizes are very close to the pattern image. For some smaller one, these these four geometric structures were fabricated using by 30 by 30 pixel image. The real size of the structure is approximately 1.5 by 1.5 millimeters. The IOP alignment is still preserved, but we could observe the severe distortion at the complex one like the star. And when we overlay the pattern image onto the structures, the structure size or the structure size are very close, as well as the bigger structures. And as mentioned, the star has severe distortion in the shape, but in the size is still close to the pattern image. And then we took the fish and the tadpole structures for further behavioral experiments. And this one we apply the magnetic field using the magnetic as well. Both of them were transferred to a new petri dish that contained PBS as the liquid medium, 1 and 2 Hertz oscillation magnetic field with 3 millitesla magnetic field strength were then applied to control the fish and the tadpole. Start by the fish on the left one, the red dot is the starting point, and the green dot is the final point of the fish. And also the same to the tadpole. The red dot is the starting point, and the green dot is the final point of the tadpole as well. We could see that both of the fish and the tadpole could be manipulated using the magnetic field, and both of them move from the place to place upward. The fish travel around 500 micrometer upward with a small shift to the right with approximate velocity of 33.3 micrometer per second, and the tadpole travel around 7 micrometer upward with a small shift to the left with approximate velocity of 35 micrometer per second. From these two videos, we could observe that different shape reports different moving behavior and also different speeds. At the final, the highlight of this work, the structure were efficiently fabricated by the Macedome, and the second is parallel fabrication of the structure would be achieved within a single batch and in a short time. The third one, the behavior of different morphologies could be immediately be investigated by the Macedome. And the last one, the magnetic functionalization of the structures was preserved due to the anisotropic alignment of the embedded IOPs. At the end, I would like to say thank you to these people to help to make this work come true. Thank you for listening.
Hello and welcome to my talk about graph-based stochastic simulations of chemical reactive systems. Let me start off by introducing the main motivation for this project. A common approach in modeling systems of chemical reactions is the following. Based on experimental data, like time traces, etc., one can infer the corresponding chemical reaction network, or CRN, which is then converted into a set of differential equations using, for example, mass-action kinetics. These ODEs can then be numerically solved for a selection of values for the reaction rate constants, and the resulting trajectories can be compared to the experimental time traces. This allows then for a way to infer the kinetic parameters by trying to fit the trajectories as close as possible to the time traces. While this is not a necessarily bad approach, judging from the fact that we can't directly observe what is going on in a test tube, it might not capture everything that is going on in the system. For example, there might be reactions that are not captured by the data due to the intermediates being hard to detect, or they happen too fast, etc. In other words, there might be something like a quote-unquote shadow network of other realistically possible reactions that are not observed due to experimental limitations. The question is therefore, which of the possible CRNs would best fit the experimentally obtained data? In this project, we want to showcase a type of modeling approach that could help answering this question. First, let me give you a quick overview of the methods we want to use in this project. The main tool that is going to be used for this project is the software package MERL. As shown on the slides, the central idea behind it is that molecules can be formalized as labeled graphs, while the reactions can be formalized as graph rewrite rules. More precisely, these rules define certain subgraph patterns within molecules and how they are transformed, reflecting the bond changes occurring in a chemical reaction. A set of initially present molecule graphs together with a set of reaction rules is then called a graph grammar. With such a graph grammar in place, one can then perform an expansion of a chemical reaction network by iteratively applying the rules to the molecule graphs. However, to prevent a combinatorial explosion of rule applications, one can furthermore define certain expansion constraints, for example, a size cutoff for the resulting new molecule graphs. Furthermore, the reaction rules themselves can be constrained by defining more or less detailed subgraph patterns. All in all, one then obtains something akin to a chemical possibility space, which is why the term chemical reaction space rather than chemical reaction network is often used. However, this is only a static approach that does not yet contain any kinetics. A way to include the dynamic behavior of a CRN is to extend MERL with a so-called stochastic simulation. As shown on the slides, this is done through a PetriNet representation where places denote molecular species holding tokens that represent their molecular counts or concentrations. The transitions, on the other hand, denote reaction events whose firing changes the token count of the in- and outgoing places, or in other words, the educt and product concentrations. A rate constant is then assigned to each transition, and the overall rate at which it is occurring is expressed using mass action kinetics, as shown on the slides. As further shown on the slides, this allows them to calculate the probability for a transition to fire, or in other words, the probability for a reaction event to occur. This is basically a typical Gillespie type of algorithm. However, due to the rule-based nature of MIL, it is possible that the network is not defined a priori, but rather expanded or explored through the consecutive application of reaction rules. This is indicated on the slides by the red part of the network. The term network on the fly Gillespie simulation is therefore often used. So in order to run a stochastic simulation, we need values for the rate constants of the corresponding reactions of the CRN. The MEL stochastic simulation framework actually has the possibility to calculate them using callback functions. The parameters that go into such a calculation can be chosen as seen fit to capture the desired mechanism to be modeled, including the molecule count of the involved species or factors that stem from the geometry of the involved molecules. However, for our purposes, we are going to use these callback functions to compute the reaction rate constants from thermochemical properties. In general, the Arrhenius law, as shown on the slides, states that the rate constant is related to the energy barrier due to the transition state that needs to be overcome. As further shown, this energy barrier can be split into a barrier height delta E between the transition state and the higher energy reaction complex and, depending if the reaction goes in the exergonic or endergonic direction, the reaction-free energy delta G. Delta G can be calculated from the formational energies of the educt and product species using external software tools. For this project, we chose the equilibrator, but any other tool can be used. Delta E, on the other hand, has to be estimated, but for simplicity, we randomized it by drawing the values from a normal distribution whose mean and variance was varied. So, with our modeling framework defined, let's have a quick look at a simple case study. The system we chose to model is the recently proposed glyoxylose reaction by Ram Krishnamurthy and Charles Liotta. It is a variation of the well-studied Formos reaction and is therefore interesting from a prebiotic chemistry and origins of life point of view. It involves glyoxylate and glycolaldehyde as initial molecules, as well as 4 reaction mechanisms: a carbonyl migration, an aldol addition and its reverse reaction and aldol cleavage, as well as a redox-neutral decarboxylation. We can formalize these 4 reaction mechanisms into their corresponding rewrite rules, as shown on the slides. To control the behavior of the simulation, the rules were formulated rather strictly. The subgraph patterns for the aldol reactions were formulated such that only linear carbon chains can be concatenated in order to prevent the appearance of unnaturally branched molecules. The subgraph patterns for the redox-neutral decarboxylation, on the other hand, was formulated to ensure that it can only occur on beta keto acids, as one usually doesn't observe it occurring on other type of keto acids. As previously mentioned, the rule-based approach of MIL can potentially lead to a combinatorial explosion in the system. In our case, the repeated application of aldol additions can lead to longer and longer carbon chains. Therefore, a further control mechanism was introduced in the form of a Hill-type function that depends on the molecule length. As we are dealing with linear chains, we take the length as the number of carbon atoms. This Hill function is then used to rescale the energy barriers for the aldol addition and cleavage, respectively increasing or decreasing them. This should capture the fact that reactions between 2 large molecules or the further growth of an already large molecule by adding a smaller one are relatively unlikely, as for longer carbon chains it is more difficult for the corresponding reaction sites to align. At the same time, the cleavage of a longer chain is made more likely, reflecting their instability. The parameters for the Hill functions were kept constant for all simulations. Let us now look at some results that were obtained from modeling the system. All the simulations were run with an initial molecule count of 200 glycolaldehyde molecules and 2000 glyoxylate molecules for 1000 time steps. Also, for each simulation, 10 repetitions were conducted and the results averaged. To get a first glance of the dynamic behavior of the system, the parameters of the normal distribution from which the values of the kinetic barriers were drawn were set to an average of 10 and a variance of 1. So in other words, rather high energy barriers with a low variation. On the slides, you can see the average time traces of the 10 species with the highest average molecule count over all runs. You can also see one of the sampled reaction networks. One can see clearly that glyoxylate is consumed, mainly due to aldol additions, while CO2 is produced and increases in concentration, mainly due to decarboxylations. To get a better picture of the dynamic behavior of the system, we looked at the time traces of the normalized molecule counts. On the left-hand side, you can see the normalized time traces for the simulation with the same parameters as previously shown. You can see nicely that some species are dependent on the formation of others, as well as that other species only arise after a relatively long lag period. On the right-hand side, you can see the normalized time traces for simulations where the parameters for the normal distributions from which the value of the kinetic barriers were drawn were set to an average of 5 and a variance of 2. In other words, lower energy barriers with more variation. You can see nicely that while the general behavior is similar to the one observed on the left-hand side, there are again species who depend on the formation of others, the dynamic behavior is rather different. Another strength of the Merle stochastic simulation framework is that it allows for the direct study of the event trace of the simulation. In other words, which type of reaction rule was sampled at what time point. By estimating the distribution of these time points, we can effectively measure the time-dependent activity of a reaction rule, which gives us another way to study the dynamic behavior of the system. The calculations of these rule activity distributions were done by using a kernel density estimation. On the slides, you can see the rule activity distributions for the simulation runs with the same parameter settings as previously shown. On the left-hand side, you can see that the system initially exhibits a higher relative activity of aldol additions. As the simulation progresses and larger molecules form, the relative activities of other rules increases as well, and the relative activity of aldol addition decreases at first, but then shows a resurgence towards the end of the simulation. Again, you can see that the system on the right-hand side, with different parameters, shows a similar general behavior, initially higher relative activity of aldol additions, accompanied by decarboxylations, both of which decrease over time. But the overall dynamic behavior is rather different. Last but not least, we can study this behavior of the system also by clustering the species by their chemical capabilities. This was done in a post-processing step where reaction smarts patterns corresponding to the left-hand sides of the Merle reaction rules were formulated and matched against the molecules of the time course. The molecules were then grouped by the amount and types of subpattern matches. This basically reflects the chemical diversity by showing in how many types of reactions the molecules of the system can be involved. As shown on the slides, we can see that increasing the energy barriers and its variability causes the system to be less chemically diverse. Finally, I want to give you a bit of an outlook of what could be the next steps to take. The work presented here can be viewed as a proof of concept study where we showed that we can use the Merle stochastic simulation engine to model the dynamics of a system, and that it is possible to ground the calculation of kinetic parameters in thermodynamics, and that one sees a change in the behavior of the system upon changing parameters like the energy barriers. For future work, the idea would be to use such a modeling framework to study real experimental data in order to conduct a form of inverse modeling as described in the beginning, but with minimal a priori assumptions. This would align with a previously proposed workflow for origins of life research, but it could be easily generalized to other fields like artificial life, synthetic biology, etc. As shown on the slides, it involves the integration of experimental results into computational models and vice versa. This way, one can have close interaction and mutual feedback between in vitro and in silico methods. I want to finish this talk by thanking my collaborators. Manuel Ulie, who is a PhD student in our group and helped out a lot with the analysis and plotting of the simulation data, Christoph Flamm, our PI, Dania Merkel and Jakob Andersen from the University of Bielefeld and the University of Southern Denmark, who are the main people behind the Merle software, Ram Krishnamurthy and Charles Liotta, whose system we chose to implement and who gave us vital feedback. I also want to thank the Novo Nordisk Foundation who funded this project and the organizers of the ALife conference to give me the opportunity to present my results here. Finally, I want to thank you all for your attention.